Hi, I'm John Graham-Cumming. I'm the author of the Geek Atlas.
So one of the places that was involved in the Human Genome Project to sequence the human genome and figure out the sequence of ATGC that we're made up of was the Joint Genome Institute in Walnut Creek, California. And it's a rather boring looking slab-sided building just off a road there, but they offer tours and it's absolutely fascinating as long as you book ahead because you get to see the equipment that is used to sequence genes today. And the actual sequencing of genes is very, very, very complicated, yet it can be broken up into a set of interesting little steps. And so what they do is they explain the work there and how they figure out the genes using a combination of biological processes and then computer power to actually figure out what the biological processes are telling you. Just to give you an example of one of the things that happens in gene sequencing, what they do is they take a piece of DNA, like something from, let's say, a virus or a human, they break it up into lots and lots of tiny little fragments, stuff those fragments into E. coli bacteria, let those E. coli bacteria breed so they get lots of copies of the DNA. And cunningly, before they do that, they've also stuffed into those E. coli bacteria two additional sequences of genes, one which gives them antibiotic resistance, and the other one which makes them fluoresce blue. And so what happens is, once they've bred all these E. coli, some of them will have actually got those genes that you're trying to sequence, and some of them won't. And the ones that haven't will not have got the antibiotic resistance, because that's inserted at the same time. So what they do is they give these E. coli a dose of antibiotics, all the ones that are useless to you, because they haven't got the genes in them, die, and then you're left with supposedly the E. coli which have got the gene you want to actually look at. Now the only problem is some bacteria survive the antibiotic without having got the gene because they've got natural resistance. So then you have a robot that looks at the ones that are glowing blue and takes those ones, says, okay, those bacteria there are worth actually looking at, and separates them out, throws away the other ones, and now they've got E. coli that contain the gene you're trying to sequence. And then what they do is they split them open and a whole other process occurs, which I could go on for about an hour about. But the people at the Joint Genome Institute do a much better job than me of explaining it. What's fascinating is a sequence of these funny little steps like that ends up with you being able to find out the 3 billion pairs of chemicals which make up a human genome, and it's a free tour. Remember to book ahead though, because it's very, very overcrowded.
I'm John Graham-Cumming, the author of the Geek Atlas, a new travel book published by O'Reilly.